pet owners used to settle for cats and dogs. Now, some labs are experimenting with gene-edited companions, micro-pigs bred for research that behave more like small pets, early efforts at hypoallergenic cats, even fluorescent fish tested in controlled environments. These aren't lining pet store shelves yet. They exist as prototypes, pilot projects, and company R&D. At first glance, it feels playful, almost trivial. But when does curiosity about glowing plants or allergy-free pets drift into rewriting life itself without limits? In this video, we'll explore breakthroughs in medicine, agriculture, and even programmable animals, and ask what happens when editing biology starts to feel like controlling time itself. The genetic time machine we didn't plan on. What scientists have stumbled upon may be the closest thing we've ever seen to a biological reset switch. Researchers in 2025 are experimenting with CRISPR-derived tools that no longer just cut DNA but act like instruments for nudging cells back toward an earlier state. These approaches don't make a person young again. Instead, they target measurable features inside cells, epigenetic marks, mitochondrial activity, and protein expression, to see whether they can shift those signals from old to young patterns in controlled lab conditions. The results so far are striking on a microscopic scale. Skin cells from older donors, when exposed to experimental reprogramming techniques, begin to behave like younger cells. They divide more efficiently. Their mitochondria generate energy more like they once did decades earlier, and proteins characteristic of youthful tissue reappear. To the researchers measuring them, the differences aren't cosmetic guesses. They can be tracked with the same precision that will be used to monitor how a wound heals. Still, these are confined to laboratory dishes and early-stage animal studies. No one has yet shown this works safely across an entire human body. That distinction matters. It is tempting to hear phrases like resetting the clock and imagine whole-body rejuvenation, but what science demonstrates today is narrow. Cells and tissues can, under controlled circumstances, be coaxed into showing markers of youth. That is promising for research into degenerative diseases or organ repair, but it does not translate into immortality or guaranteed lifespan extension. The leap from rejuvenating cells in a dish to rejuvenating people remains immense. The rise of these tools comes in parallel with genetic medicine entering real healthcare. In 2025, a 1-year-old boy named KJ at the Children's Hospital of Philadelphia received what has been described as the first personalized in vivo CRISPR treatment for a metabolic disorder called CPS1 deficiency. His therapy directly edited his liver cells inside his body, something that a decade ago seemed impossible. This was a milestone proof that CRISPR-based methods can reach patients safely. But that case treated a lethal single-gene disorder. It did not attempt to reverse aging. Mentioning it here highlights how gene editing is making its way from experiments into clinics, while anti-aging applications remain firmly experimental. So far, the most persuasive data comes from looking at how epigenetic markers, the chemical tags that tell genes when to switch on or off, change as cells grow older. By nudging these markers back toward patterns seen in youth, scientists can measure functional improvements, like a cell processing energy more efficiently or repairing damage more effectively. Put simply, they can now measure a cell's biological age, and in some cases adjust it backwards. But to keep perspective, making cells younger in a lab does not yet mean making a person younger. The possibilities still capture imagination. A therapy that could rejuvenate sections of tissue might one day help hearts weakened by age, livers scarred by disease, or neurons at risk of degeneration. Even for organ transplantation, the ability to renew an old organ for reuse could revolutionize medicine. These scenarios are speculative, but the early cellular results point toward them. At the same time, the questions multiply. If a 70-year-old's cells could be coaxed back toward the condition of a 30-year-old's, would their organs last longer? Could cognitive decline be slowed if neurons were reset? No one knows. The mechanics of rejuvenating scattered cells versus orchestrating a safe, body-wide intervention are vastly different challenges. Undoing an epigenetic mark in a cell culture does not guarantee that a living system with immune defenses, complex feedback loops, and cancer risk can tolerate the same intervention. And as history shows, breakthroughs rarely roll out evenly. When gene editing first emerged, it was confined to a few labs. Within years, it was widely adopted in agriculture and medicine.
If cellular clocks behave the same way, they may first appear as specialized, prohibitively expensive
Therapies, accessible only to a handful of patients. The technology itself could become a divider, separating those who can afford biological youth from those who cannot. In such a world, age might stop being just a number and start being another marker of inequality. This tension is underscored by how different cellular rejuvenation is from conventional approaches. Vitamins, hormones, or anti-aging creams have always targeted surface effects. Fewer wrinkles, stronger bones, slightly extended health. By contrast, resetting a cell's biological age at just the very instructions cells follow. That is not maintenance, it is rewriting. It is what makes scientists describe these tools not as bombs that soothe damage, but as clocks that can, in small increments, be moved backwards. It is easy to understand why this captures attention. Beyond everyday medicine, imagine preparing astronauts for long missions where radiation steadily ages their cells. Being able to periodically reset some of that cellular wear could mean safer travel. Or imagine patients with degenerative diseases where rejuvenated tissues might delay decline. Every example piles more weight on the same fundamental question. If we can make some cells young again, should we try to apply that power as widely as possible, or reserve it for the most urgent medical needs? This dilemma is the heart of the debate. Using such tools to prevent early onset disease seems justifiable. Extending healthy lives for those already old begins to push into territory societies have never experienced. Our institutions, from retirement systems to cultural rhythms, are built on the arc of age that everyone eventually follows. Changing that arc destabilizes not only biology but also economics, family structures, and collective expectations. Meanwhile, for every exciting result, there are reminders of the risks. The same cellular rejuvenation pathways that restore vitality can, if uncontrolled, produce runaway growth. That is essentially the definition of cancer, a cell that forgets its boundaries. Rejuvenation experiments must therefore be balanced against the danger of triggering malignancy or dangerous autoimmune reactions. The line between repair and harm remains narrow, and no one pretends it is easy to navigate. Investors nevertheless see extraordinary potential. Aging is a universal condition. A proven method to slow it would touch every family in every country. That kind of impact attracts massive funding. Startups are already formed on the promise of shepherding rejuvenation techniques into clinical trials, and private capital is flowing in anticipation of one of the largest markets ever imagined for biotech. The science is still uncertain, but the hope and the potential reward drives growing momentum. For now, though, progress is confined to careful, incremental experiments. Scientists publish results showing they can track cellular age, reverse marks in limited settings, and restore certain functions. Each step underscores the same message. Time within biology may have more flexibility than once thought. That is not immortality, but even a small degree of control changes how medicine views health and decline. The story behind the headlines is quieter. It is about fine-tuning enzymes, calibrating guide RNAs, adjusting transcription factors, and logging tiny yet consistent changes in cell states. No one is promising overnight cures, but even careful, incremental success reframes questions once buried in philosophy. If cellular age can be reset in the lab, who should decide when and how that ability is applied? Do we use it to treat the sick? To extend the healthy? Or to reinforce social divides? The experiments themselves cannot answer that. And one more tension remains. As extraordinary as the idea of reversing age is, many of the most urgent uses of gene editing are not about people at all. They concern the food systems we rely on every day. Long before rejuvenation therapies reach society, the same tools are already being tested on the plants that fill our grocery stores. The stakes there are less about living forever and more about whether some of the most familiar foods on our plates will survive into the next generation. That shift in focus sets up a striking contrast. On one side, laboratories probing the limits of human biology. On the other, fields where genetic tools may determine whether entire crops endure or collapse. Both hold the same underlying question: when we can edit life, who decides how far to go, and for whose benefit? Bananas, extinction, and the gene editing gamble. Walk into almost any grocery store and you'll find bananas lined up in perfect rows, each one looking nearly identical. That uniformity isn't an accident. Almost every banana sold for export belongs to the same variety, known as the Cavendish. Its smooth peel, even ripening, and mild sweetness made it ideal for shipping across oceans and appealing to consumers unfamiliar with tropical fruit. But the very genetic sameness that made it successful has also turned it into an extremely vulnerable monoculture. Unlike apples or grapes, where different varieties compete, the global banana trade relies overwhelmingly on a single clone. This vulnerability has collided with a disease called tropical race 4, or TR4, a soil-borne fungus also known as Panama disease. The fungus infiltrates the roots of the plant, blocking water and nutrients, and eventually killing the banana tree. Once TR4 is in the soil, it can remain for decades, making the land unusable for Cavendish bananas. No fungicide can wash it away. In the early 2000s, the disease spread beyond Southeast Asia, reaching the Middle East, Africa, and eventually Latin America, the world's last stronghold for banana exports. By 2019, confirmed outbreaks in Colombia signaled that even South America was no longer safe. For the banana industry, the stakes are staggering. The export trade alone is worth an estimated $12 billion annually, and it supports the livelihood of millions of farmers in nearly 50 countries. For local communities in Africa, Latin America, and Asia, bananas and plantains are more than commodities. They are daily staples, providing affordable calories and reliable nutrition. Losing the Cavendish isn't just about shoppers in London or Los Angeles missing a favorite fruit. It could mean food insecurity and economic collapse for families who rely on bananas both as income and as their main source of sustenance. Given the scale of the threat, the obvious question is why the world can't simply move on to another type of banana. Thousands of banana varieties exist, some resistant to TR4, but most bruise easily, ripen too quickly for shipping, or have flavors unfamiliar to consumers. The Cavendish became dominant not because it was the most delicious or resilient, but because it could travel long distances, ripen predictably, and meet the expectations of a global market. Entire infrastructures of production, logistics, and distribution were built on this single fruit. Switching varieties would mean rewriting decades of trade systems, retraining farmers, and convincing consumers worldwide to accept a fruit that looks and tastes different, and none of that has proven easy. That is why scientists have turned to CRISPR and related biotechnologies. Traditional breeding, the method often used to develop resistance in crops, fails here. Bananas are seedless, and they don't reproduce in conventional ways. Attempts to cross Cavendish with resistant wild relatives have largely produced inedible results, full of seeds or lacking sweetness. CRISPR offers an alternative by letting researchers directly edit the Cavendish genome, copying natural resistance traits found in other bananas without altering the fruit's familiar taste or shipping resilience. Field work in Australia, led by Professor James Dale, has produced promising TR4-resistant lines, some developed through cisgenics by introducing banana genes from resistant relatives and others using new editing methods. Companies have also joined in. Elo Life Systems, a biotech firm, launched field trials of gene-edited Cavendish in Central America in collaboration with Dole, aiming to engineer resistance through targeted proteins that make the plant inhospitable to TR4. Meanwhile, the Swiss-Indian company Initerra is cultivating Cavendish mutants that survived TR4 outbreaks in the Philippines, selling them under names like InnoGreen and InnoBlack, with the hope they retain the reliable flavor and texture the global market demands. Each of these efforts underscores the urgency. TR4 is not slowing down, and time is running out to keep the Cavendish alive. Early attempts at editing showed the risks of tinkering too much. Some modified bananas developed chalky textures or sour-off flavors, traits that made them commercially useless even if they resisted disease. The challenge was to preserve the qualities that global trade depends on: shelf stability, transport durability, predictable ripening, while adding fungal resistance. By 2025, several efforts finally reached large-scale field testing in infected soil. Reports from those trials indicated that edited banana plants managed to survive where unaltered Cavendish quickly withered, keeping leaves green and roots healthy in conditions known to wipe out conventional crops. For farmers, this difference could mean enduring income instead of abandoning their fields forever. Yet the technology faces hurdles that are not scientific but political and cultural. Regulatory approaches vary widely. In the United States, Japan, and several other countries, gene-edited plants that do not introduce foreign DNA are often subject to lighter regulation than older genetically modified crops. But in Europe, where consumer skepticism of GMOs remains strong, rules treat edited crops with the same degree of scrutiny, making commercial adoption slow and unpredictable. This divide means the same banana could be approved for sale in one region but banned in another, complicating global trade. Even if regulators agree, consumer perception could prove decisive. Bananas are among the most popular fruits worldwide, and the Cavendish as it exists today is already a human-made crop, propagated exclusively by cloning. But many buyers still draw a sharp line between cultivated agriculture and direct genome editing. A CRISPR-engineered Cavendish may look, peel, and taste exactly like the fruit millions already eat, but suspicion of the label genetically modified could still deter shoppers. For retailers wary of backlash, even the perception of tampering might discourage stocking edited bananas regardless of their necessity for survival. The cultural stakes echo far beyond breakfast tables. In countries where bananas make up a significant portion of the diet, resistant varieties are urgently needed. Without them, TR4 can trigger shortages that ripple through food systems, increasing hunger in vulnerable regions. Biotech solutions represent one of the few clear technical answers to this problem, but whether governments and populations accept those solutions remains uncertain. Some see the Cavendish crisis as a test case. If the world embraces gene editing here, it might open doors to saving other crops threatened by drought, pests, or climate stress, from rice to maize to wheat. If resistance to editing prevails, then some of those crops may become casualties of the same rigid stance. The banana is thus more than just a crop at risk. It is a measure of how far societies are willing to go in rewriting the genetics of the plants that feed them. What happens next hinges on how TR4 continues to spread and how persuasive edited bananas prove in the field. If the disease keeps advancing, the global supply chain faces collapse within a generation. If edited or cisgenic Cavendish lines survive, the fruit could have a future, but only if governments approve them and consumers accept them. In other words, the science may already be providing answers, but whether those answers reach supermarket at scale depends on public trust and political will. If TR4 keeps burning through plantations without a viable defense, Cavendish bananas, the variety most of the world knows, may disappear from shelves entirely. Gene editing offers one of the only technically scalable ways to prevent that collapse. But the success won't be determined just in laboratories or plantations. It will be determined in how societies weigh the value of preserving a food staple against their doubts about biotechnology. This tension between a technology that could avert a collapse of a familiar fruit and the skepticism that keeps it from being widely adopted serves as a reminder. The same tools used to protect bananas are increasingly being applied to far more mobile, less predictable organisms. In some places, those organisms are not staple foods, but living creatures that move through neighborhoods, breed on their own, and transform environments in real time. The consequences of editing them unfold not in plantations, but out in the open, where people live. Mosquitoes are now programmable life forms. In some towns in Florida and across trial regions in Brazil, mosquito control no longer relies only on insecticide sprays or draining pools of standing water. Instead, it includes the planned release of living edited organisms. These are mosquitoes reprogrammed at the genetic level to behave in precise ways, as though their instructions were written into code. The idea is straightforward. Modify the mosquito before it enters an environment, and the insect itself becomes the vector of a public health strategy. In practice, this often means releasing edited males that do not bite, designed so that their female offspring never survive beyond a juvenile stage. Over time, local populations shrink without further human intervention. Unlike chemicals that need repeated application, these organisms propagate their genetic instructions on their own. The diseases these experiments target remain some of the most dangerous in the world. Dengue fever spreads explosively through crowded cities. Zika appeared suddenly and raised fears of birth defects. Malaria's toll continues at a scale of hundreds of thousands of deaths each year. Standard interventions have struggled to keep pace. Insecticide use breeds resistance. Bed nets offer partial protection but fail to eliminate disease, and vaccine distribution remains limited. Faced with this cycle, the question became whether the mosquito itself could be altered so that it either could not transmit pathogens or could not reproduce in a way that sustains outbreaks. That possibility became attainable when gene editing allowed engineers to design inheritance patterns that bypassed normal genetic randomness. This is where the concept of a gene drive enters. Normally, a genetic trait has a 50-50 chance of being passed down, making its survival uncertain across many generations. A gene drive is engineered so that the altered trait is inherited almost every time, removing that natural randomness. In theory, releasing a relatively small number of edited insects could result in entire populations carrying a new trait within a handful of breeding cycles. It is a method that makes insects function less like random breeders and more like self-replicating programs. Evidence has been emerging not only from field introductions but also from detailed laboratory work. A 2025 study, carried out by researchers at institutions including UC San Diego and Johns Hopkins, demonstrated a striking genetic change in Anopheles mosquitoes. By editing a single amino acid in the FREP1 protein, which malaria parasites rely on to reach the insect's salivary glands, the scientists effectively blocked parasite transmission. Mosquitoes carrying the altered allele had dramatically fewer infected parasites in their glands, often none at all. The study went further, showing that this resistant variant could be driven through laboratory populations using an allelic drive, a system designed to spread a natural allele across the group until nearly all individuals carried the protective form. This offered proof that traits designed to limit disease transmission could not only exist stably in the insects, but could be encouraged to dominate entire populations in controlled experiments. Small-scale field projects have already extended these principles outside laboratory cages. In Florida and Brazil, carefully approved and monitored trials have used edited male Aedes mosquitoes designed so that their female offspring do not mature. These projects remain in early phases, limited in numbers, strictly regulated, and under constant monitoring. Years away from anything that could be considered a global deployment, they nonetheless provide tangible evidence that engineered traits spread reliably in real-world conditions. Traps and release areas have detected edited males and their genetic signatures persisting across several reproductive cycles, supporting models that predicted rapid penetration of a designed trait into local environments. Would you accept engineered mosquitoes being released near your home if the goal was to end malaria or dengue? For some, the answer is yes, if the cost is fewer funerals and fewer children falling ill. For others, the answer remains no, because the long-term ecological consequences cannot be reversed once such organisms are established. This tension defines the current moment, the balance between saving lives immediately and guarding against environmental changes that may unfold too slowly to be seen until years later. Communities in test zones have voiced both hope and apprehension. Families burdened by repeated bouts of dengue embrace the idea of fewer fevers, lighter strains on hospitals, and an end to cycles of illness that drain household incomes. At the same time, residents ask who authorized their neighborhoods to act as live laboratories. Bioethicists stress that projects cannot succeed without real consent and engagement. As Natalie Koffler and other voices have pointed out, protocols for genetically modified organisms have historically fallen short in ensuring affected communities were fully informed. Without transparency, any promise of health gains risk-building mistrust. Beyond social consent, ecological questions linger. Mosquitoes feed birds, bats, and fish. Eliminating or suppressing them may create unanticipated consequences throughout food webs. Some researchers argue that reduced mosquito abundance would barely affect predators, pointing to dietary flexibility. Others caution that tampering with one component of an ecosystem can ripple outward in ways that models rarely capture. Concerns intensify when considering an approach like a gene drive, which is designed to be permanent. Unlike a pesticide that degrades in weeks, or a net that can be withdrawn overnight, a genetic change spreads forward indefinitely once introduced. That permanence is what makes mosquito editing both promising and unsettling. There is also the practical issue of borders. An insect released in one jurisdiction may not remain there. Mosquitoes can spread naturally or be carried by commerce to neighboring regions, meaning that one country's decision could impose consequences on others without their agreement. This raises questions of sovereignty that parallel, but also exceed earlier debates about genetically modified crops. Crops can be confined behind fences, insects cannot. The discussion is no longer about one laboratory or one national program. It becomes a transnational fact when the insects themselves are mobile. The trials happening in 2025 mark only the beginning of what advocates imagine. Public health models envision regional suppression of Aedes aegypti, reducing dengue transmission to negligible levels. They predict malaria transmission collapsing once resistant variants take hold across Anopheles populations. In terms of potential benefit, the numbers are staggering. Hundreds of thousands of lives saved yearly, billions in healthcare costs avoided. Yet conservation scientists and policymakers urge slower steps. Researchers emphasize phase release, rigorous ecological study, and precisely defined consent processes for each community affected. Without them, the technology may face opposition strong enough to halt progress. Reactions in trial sites reflect this divide. Some households report dramatic improvement. Evenings outdoors feel different when children surface with far fewer bites, when clinics see fewer patients, and when there is a respite from the constant anxiety of mosquito-borne illness. On other streets, skepticism remains. Residents question logs of trap counts, worry about silent ecological shifts, or suspect that corporations use their neighborhoods to test profitable technologies under the label of public service. For them, the idea of fighting disease by adding insects rather than taking them away remains counterintuitive, even alarming. The global community watches closely. If trials continue to show reductions in biting pressure and disease incidents, pressure will grow to expand such programs into malaria-endemic regions of Africa and Southeast Asia. The debate will then circle around not only ecological risk but also economics. Who will own the engineered lines? Will their deployment be treated as a universal public health tool or as a licensed product controlled by patents? Access, affordability, and governance could determine whether these tools are used where they are most needed or remain confined to nations that can afford them. For now, what stands out is the reframing of mosquitoes themselves. Once considered the most familiar pests, they have become templates for programmable life. Each engineered edit represents a line of human-written code living inside an animal that mates, flies, and spreads it freely. This transformation captures both the extraordinary promise and the unsettling permanence of the gene-editing age. For societies battling diseases, the hope is undeniable. For those weighing ecological balance, the risk remains sober. These tensions between improvement and uncertainty, between saving lives and maintaining trust, are reminders that biotechnology no longer applies just to plants in fields or cells in laboratories. It also applies to creatures that share our environment and rewrite themselves at the genetic level according to instructions we have authored. And this shift, where familiar organisms become luminous signals of what editing can achieve, leads naturally to the next frontier, a very different kind of engineered organism, designed not to bite or transmit disease, but to glow. Glow-in-the-dark plants from Mars first, your desk next? Glow-in-the-dark plants sound like something out of a science fair or a design catalog, but the research taking place in 2025 shows that they are being considered for serious, albeit early-stage applications. Scientists and students are experimenting with ways to use bioluminescent traits not as gimmicks but as tools, potential sources of low-energy light and real-time stress sensors in environments where traditional systems are heavy, costly, or fragile. Projects backed by NASA and the ISS National Lab, such as the Genes in Space competition, highlight the growing interest in genetic tools that could one day help keep crops alive during long missions. These efforts remain exploratory, not core survival technologies, but they illustrate how gene editing is being tested in contexts where every gram and every watt matters. One of the biggest challenges in orbital or planetary agriculture is that plants behave differently when normal environmental cues vanish. On Earth, roots grow downward under gravity while shoots arch upward toward light. In microgravity, those signals blur, and plants often twist unpredictably or misdirect energy. Add in radiation damage and the stresses of closed habitats, and traditional growth strategies break down. Researchers looking at light-producing traits see a potential way to make plants less dependent on external systems. Instead of relying on cargo lamps or wiring arrays, they imagine organisms that produce a faint internal glow. For astronauts scanning a small greenhouse, that glow can also become a quick visual indicator of stress. When a plant glows less brightly, it signals trouble before an in-depth test is needed. The key to creating such self-illuminating plants has been importing genetic elements from naturally glowing organisms, such as marine bacteria or fireflies. Using CRISPR, scientists have spliced in genes that allow seedlings to produce their own light. Early versions shine weakly, more like a faint shimmer than a lamp. But those glows respond to the plant's internal conditions. Differences in brightness map onto stress, hydration, and metabolic state. In cramped, resource-limited environments, such a built-in communication system has obvious advantages. Instead of running elaborate diagnostics, crews could look across a row of plants and see, in color and intensity, a snapshot of their overall health. For environments like Mars habitats, every minute saved and every sensor not carried from Earth makes a difference. Yet it is important to recognize that these remain speculative prototypes, not operational assets of space agencies. While popular illustrations depict glowing farms lighting entire domes, the reality is far less dramatic. Current iterations produce dim light that is unsuitable for reading or photosynthesis but useful for signaling. And there's a cost. Producing light drains metabolic energy that the plants would otherwise use for growth and survival. Striking a balance, bright enough to be useful as an indicator without undermining the crop, remains a central technical hurdle. Researchers acknowledge that for now these organisms are testbeds for genetic methods, not mission-critical agricultural tools. The Genes in Space program gives a sense of how such experiments fit into broader space biology research. In 2025, for instance, student scientist Nietzsche Joer won for her work on codon usage bias in microgravity, a project not directly about glowing plants, but about how genetic information is interpreted differently under space stress. Past milestones have already shown that genomic technologies work off-world. NASA astronaut Kate Rubin's DNA aboard the ISS, and ESM Peak ran the first PCR reaction in orbit. Together, these projects build a foundation. If scientists can sequence, edit, and monitor genes in space, then engineering glowing indicators becomes another step in the same trajectory. The theme is not so much that they will illuminate Martian farms soon, but that they represent part of an expanding toolkit for controlling and monitoring biology under extreme conditions. There is also an earthly angle. Startups see potential novelty markets in houseplants that glow softly in the dark. Imagine replacing a nightlight with a potted plant, one that emits a green or blue hue. Investors imagine positioning such products as premium decor that doubles as conversation pieces, blending sustainability pitches, plants that consume carbon dioxide while emitting glow, with luxury branding. This market is not yet mature, and the organisms available are fragile prototypes confined to labs, but the diversity of interest, from NASA engineers to design-focused biotech firms, shows how gene editing can create products that straddle utility and novelty. Beyond homes, some researchers speculate about glow traits for low-resource scenarios. Rural communities, disaster relief zones, or refugee camps often lack stable power grids. In theory, distributing seeds of plants that glow faintly could provide short-term illumination without batteries or wiring. The light would be dim but enough to mark paths or organize enclosed shelters at night. These scenarios are highly speculative, and no one is planting glowing crops in 8 operations today, but the prospect underscores how living systems could take on utility currently reserved for manufactured equipment. In each case, space stations, Mars habitats, or remote villages, the appeal is that organisms carry their own functions instead of requiring imported infrastructure. Still, technical barriers are substantial. Bioluminescence genes are energy-intensive. Too much expression, and plants slow growth or fail to thrive. Too little, and the glow is useless. Different genetic circuits have been tried to regulate when and how light is produced, but finding an optimal balance is painstaking, requiring trial and error over many generations. On top of this, radiation in space or harsh soil conditions on Mars could destabilize engineered processes. Glow may not persist consistently across those stresses, making the trait unreliable. For commercial use on Earth, regulation poses an additional barrier. Biotech firms face fragmented rules. Some countries treat gene-edited organisms under lighter law. Others apply the strict categories once assigned to first-generation GMOs. A decorative glowing ivy might be legal in one market but banned in another. Intellectual property further complicates matters. Bioluminescence genes are often patented, as are the circuits used to regulate them. If glow traits become marketable, their spread could mirror seed patents in agriculture, where a few companies control access and pricing. In luxury markets, this may be accepted as branding. But if such traits ever move toward wider public uses, questions of affordability and access will emerge. Whether engineered biology should be treated as equipment, as medicine, or as consumer decor remains unresolved. Despite these challenges, the imagery of glowing crops on extraterrestrial colonies has power. Renderings circulate that imagine Martian domes bathed in biological glow, framing survival not as a sterile technological feat but as a partnership with modified nature. These renderings exaggerate existing science, but they also anchor public imagination. They give a glimpse of how gene editing can blur lines between aesthetics, function, and survival. In this sense, glow-in-the-dark plants symbolize both the ambition and the ambiguity of CRISPR-driven innovation. They are not food or medicine, but something stranger, a coded adjustment that turns organisms into multi-purpose tools. This raises cultural and ethical questions. What does it mean when plants are designed to act as sensors or lamps? People have long accepted ornamental breeding for color or fragrance, but integrating luminescence from microbes crosses into creating hybrids that never existed in evolution. Do we begin to think of plants less as life and more as devices? Does altering their function in this way erode the cultural meanings we attach to greenery as symbols of growth, stability, or connection to the natural world? These are questions technology cannot resolve on its own. For now, glow-in-the-dark plants remain laboratory novelties with possible futures branching in several directions. They may emerge as consumer decor in urban apartments. They may never reach mass use at all if energy efficiency or public acceptance barriers remain too high. But they illustrate clearly how genetic engineering is pivoting toward roles that exceed traditional categories. The shift from food and medicine to environment-shaping organisms demonstrates just how far the editing lens has widened. And as with other frontiers of gene editing, these organisms invite us to ask not just whether we can design them, but whether we should. Do we turn plants into tools and sensors, and if so, does that change how we see and value them? These reflections echo outward because the next set of applications pushes genetic engineering into another everyday system that humans depend on, our global food supply, and the organisms at its center influence the atmosphere itself in ways most people rarely notice. Cows that burp less methane. Number 1. Of the clearest places where gene editing intersects with climate change is in livestock. Among the gases warming the atmosphere, methane stands out for its intensity. While cows may not look like factories, their stomachs act like microbial engines that generate large volumes of methane with every meal. Burps from more than 1 billion cattle worldwide now add up to a climate footprint larger than many industrial sectors combined. On a per-animal basis, the average cow releases about 220 pounds of methane each year. Methane remains in the atmosphere for less than 2 decades, but during that span it traps far more heat than carbon dioxide. This makes it one of the fastest levers available for slowing near-term warming if the source can be changed. Scientists have approached the problem in 3 main ways. Some focus on modifying diets, adding compounds like 3-nop or small amounts of red seaweed to lower methane production in the rumen. Trials have reported reductions of 30% or more, with some seaweed experiments showing greater than 60% cutbacks. But scaling that approach is difficult. Additives are expensive to supply at global herd scale, variable in quality, and often impractical where cows graze freely on pastures rather than controlled feed. A second direction involves selective breeding, identifying naturally low methane cattle and adapting herds across generations, a method slower to deliver results. The third and perhaps most ambitious strategy aims to reprogram the root cause, the microbes inside the cow's rumen that actually produce methane. The rumen, a fermentation chamber that functions as the cow's largest stomach compartment, hosts a dense network of bacteria and archaea. These microbes specialize in breaking down cellulose, the tough fibers in grasses. Without them, cows could not survive on their usual diet, but as they ferment that plant material, some species release methane as a metabolic byproduct. Under normal circumstances, the gas builds up in the rumen and is expelled by the animal through burping. Over evolutionary time, this trade-off made sense. Cattle gained access to energy locked in grasslands, and methane released into the air was negligible. Scaled up to the modern livestock industry, however, the same process amplifies into a planetary problem. This is where researchers at the University of California, the Davis, working with the Innovative Genomics Institute, have set their sights. IGI researchers are now pursuing microbial genome editing to cut methane at its origin point. By sequencing the DNA of methane-producing microbes, they have identified the genes that drive the emissions. Using tools like CRISPR-Cas9, the goal is to make precise edits that remove or replace those instructions without disturbing the microbe's other functions. In principle, the reprogrammed microbes perform the same digestive work but no longer vent methane as a side effect. The ambition is to deliver this as a one-time probiotic treatment during calfhood, reshaping the microbial community for the animal's lifetime. The early laboratory findings suggest this approach could work. In controlled environments, shifting microbial populations lowered methane output significantly, sometimes approaching reductions in the 40% range. Models suggest a scaled version of this could be a powerful lever for near-term emissions cuts. But scaling comes with hurdles. Microbes edited successfully in a lab dish must survive inside real cattle, interacting with a complex ecosystem of trillions of other species. It remains uncertain whether engineered strains can persist long-term, or whether the existing microbiome will outcompete them. Field trials in living herds are the next step in answering that question. Dietary additives remain part of the conversation precisely because they have delivered dramatic experimental results. Red seaweed trials, in some cases, cut emissions by up to 2/3. Still, costs for cultivation, transportation, and distribution are high. Farmers would need a constant supply of the additive, delivered consistently in feed, something impractical for smallholders or for cattle grazing freely. Vaccines against methane-producing microbes have been tested too, aiming to lower their population selectively, but results were inconsistent. These techniques demonstrate potential but often collapse under real-world economic and logistical pressure. Microbiome editing differs by aiming for permanence, alter the biology once, and the animal maintains the trait without constant intervention. What makes this research distinctive is how it redefines the boundary of what counts as genetic editing in livestock. Instead of altering the cow's own DNA, scientists focus on the microbes it carries, organisms just as essential to its digestion as the cells of its stomach wall. In this view, adjusting the microbiome resembles upgrading a probiotic more than redesigning the cow itself. If restructured, microbes successfully colonize a young calf. They replicate within the gut for life, passing to future generations through natural microbial transfer. The animal grazes, lactates, and gains weight as before, while its emissions profile quietly shifts. Farmers would see the same animals performing in the same ways, with invisible changes embedded in the microbiology of digestion. Yet technical hurdles are only half the equation. Public trust in consumer acceptance may prove decisive. Even though the editing targets microbes rather than the cattle themselves, skepticism about genetic modification remains widespread. Milk and meat from such animals could encounter resistance in regions with strict GMO regulations, particularly in Europe. Labeling debates, import restrictions, and trade friction are predictable outcomes. Investors and researchers at IGI are aware of this and present the approach explicitly as a probiotic update, not as fundamental genetic alteration of livestock. Whether that framing convinces consumers remains uncertain. The ethical debate continues over whether this focus solves the right problem. Critics argue that the real challenge is the sheer number of cattle raised for consumption. Beyond methane, herds contribute to deforestation, water use, and land conversion for feed. Adjusting microbial emissions per animal, they contend, does nothing to address the scale of consumption itself. Instead, it risks offering a technological fix that maintains high demand for beef and dairy while postponing shifts toward more sustainable diets. Supporters counter that dietary change across billions of people is unpredictable and slow, tied to culture, livelihoods, and national identity. A tool that lowers emissions while keeping habits intact is, in their view, both pragmatic and urgent. These are not mutually exclusive approaches. In practice, both may develop side by side, demand gradually shifting downward while biotech solutions mitigate the short-term footprint. The coming years will test whether edited microbes can survive real cattle conditions. Researchers are preparing trials outside the lab, where competition among microbial strains is fierce and daily feed varies. Success will be measured not just in emissions data, but also in whether animals remain healthy, productive, and indistinguishable from untreated cattle in taste and nutrition of their products. If proven safe and effective, a one-time calf treatment could spread quickly across different systems, from industrial feedlots to smallholder herds. The simplicity of a permanent probiotic replacement appeals precisely because it does not require constant oversight. A solution that scales evenly across rich and poor farming systems alike could have a global reach. Still, the social and regulatory environment may be the bottleneck. Consumers opposed to genetic modification may require compelling proof before accepting products tied to microbiome engineering. Governments hesitant with gene-edited crops may apply similar caution to livestock, extending approval processes across years. The success of IGI's treatment, therefore, depends not only on biology, but also on narrative, whether the public sees this as helpful probiotic engineering or as another form of genetic manipulation entering their food chain. The tension brings the story back to a core theme running through many corners of gene editing. In theory, the science creates elegant levers to reduce risk or improve resilience without forcing dramatic lifestyle changes. But the power of those levers depends on public trust and the willingness of societies to accept biology that carries human code. Cattle burps illustrate the paradox acutely. The methane problem is real, and gene editing might cut it at the source. Whether that possibility becomes reality is a question of governance and perception as much as genetics. The promise is that edited microbes could cut livestock emissions without changing what people eat or how farmers raise animals. The uncertainty is whether societies are willing to accept organisms reprogrammed at such an intimate level within the food chain. That unresolved question points toward broader frontiers, where editing life takes place in environments even less forgiving than a pasture or dairy barn. There, the stakes of genetic survival play out under conditions that Earth's biology never evolved to handle, and where the rules of life itself begin to feel different. Gene editing in outer space. The environment of space reshapes biology in ways Earth never prepared us for. Life evolved under the steady pull of gravity and beneath atmospheric shielding that filtered out much of the radiation streaming through space. Remove those protections, and organisms begin to reveal vulnerabilities and responses we rarely see at home. Microgravity alters how fluids move through tissues, while cosmic rays slice into DNA with a frequency no Earth-based model can fully mimic. By 2025, researchers have begun treating orbit not just as a travel frontier, but as a laboratory, sending gene-edited organisms beyond Earth to examine how life withstands stresses that cannot be recreated perfectly on the ground. The current subjects are not humans but microbes and yeast, organisms with short lifespans, fully mapped genomes, and easily observed cellular behaviors. In sealed containers aboard the International Space Station, edited strains share space with unedited controls, facing daily bombardment from radiation that often fractures DNA. On Earth, experiments predict strand breaks, mutations accumulating, and proteins failing from damage. Yet in orbit, some edited lines display surprising resilience. Certain yeast strains seem to repair broken DNA strands faster or activate backup repair pathways rarely triggered in terrestrial labs. These unexpected responses suggest that space may act as both a hazard and an adaptive trigger, an environment that forces biology to reveal hidden options. This line of testing builds on a foundation already in place. The space station has hosted major milestones in molecular work. Astronaut Kate Rubin sequenced DNA in orbit. Tim Peake demonstrated PCR reactions, and student-led projects under the Genes in Space program have explored CRISPR activity in microgravity. These breakthroughs demonstrated that sequencing and gene editing reactions function aboard the ISS, setting the stage for current microbial studies that push from proof of concept toward targeted adaptation. The very fact that DNA sequencing and CRISPR interventions practical in orbit confirms that gene editing research does not need to stop at Earth's edge. Why microbes matter becomes clearer when considering missions measured not in weeks but in years. A trip to Mars or a semi-permanent lunar base exposes astronauts to relentless radiation and bone-weakening microgravity. A small improvement in DNA repair, multiplied across countless cells, could spell the difference between long-term viability and chronic decline. Microbes serve as simple test cases, stand-ins for how more complex systems, crops, gut flora, or even human tissues might benefit from similar edits. If resilience-enhancing variants succeed at this microscopic scale, the logic extends to food security and crew health, offering building blocks for sustainable life off-world. Radiation remains the major obstacle. Astronauts outside low Earth orbit face doses far exceeding occupational limits at home, heightening risk of cancer, neurodegeneration, and circulatory disease. Traditional shielding can only reduce the dose so much, and physical barriers add prohibitive mass to spacecraft. This points toward a different strategy: enhance the biology itself. Research papers in reuse have proposed testing base editing and prime editing technologies in microgravity to correct damage more efficiently. Others suggest borrowing genes from extreme-tolerant organisms, such as the DNA-protecting proteins of tardigrades, that could bolster human cell resilience. These remain long-term ideas, theoretical and requiring intensive safety checks, but observations of altered DNA repair in orbit hint that biology may already contain underutilized tools. Equally disruptive is microgravity's effect on physiology. Our cells rely on mechanical cues, pressures, tensions, orientation in a gravitational field to regulate division and growth. Without those benchmarks, cellular functions shift. Astronauts experience muscle atrophy, bone density loss, immune suppression, and even ocular changes as fluids redistribute unnaturally. In this context, gene editing becomes more than radiation defense. It becomes a potential way to modify how organisms maintain themselves in reduced gravity. Skeletal muscle loss could be addressed not only by exercise devices, but by edits that amplify protective pathways in muscle cells. Bone density might be preserved by sustaining osteoblast activity beyond normal regulation. These remain speculative, but they align with current studies examining how edited microbes or plants react differently in weightless environments. Practical tests are already moving forward with probiotic microbes. Gut microbiomes shift in space, sometimes reducing efficiency and immune support. Efforts in 2025 explore whether edited strains of bacteria can establish stable, productive colonies in orbit that resist these stresses. A reliable probiotic capable of producing vitamins and maintaining immune balance would lessen reliance on regular shipments from Earth. A capsule containing engineered bacteria could replicate across missions indefinitely, turning a single dose into a renewable supply. The lesson is that small edits to microbes can have outsized effects on astronaut health and logistics. Plants bring food security into the conversation. As cosmic rays damage plant DNA, harvests can falter. Traditional breeding cannot outpace constant particle bombardment, but genetic interventions may enhance DNA repair or build resilience into crop genomes. Experiments have already shown that edited seeds sprout and grow more consistently than controls in orbit. If edits can consistently protect plants, crop reliability becomes more feasible for Mars greenhouses or lunar stations. Edits could also fine-tune nutritional profiles, designing plants richer in protein or vitamins suited for closed diets. Agriculture in space represents the line between fragile outposts and self-sufficient colonies, and editing genomes may help cross it. Still, the work comes with deep ethical questions. Should humans edit themselves for space, or should environments be designed to accommodate unaltered biology? Some futurists argue genetic modification might eventually be essential for colonists to thrive in radiation-heavy or gravity-light settings. Yet these ideas remain theoretical proposals, controversial and ethically fraught. Critics like former NASA bioethicist Paul Root-Wolpe warn that tampering with the human genome to fit space may undermine what it means to be human. Such alterations, he argues, risk unforeseen consequences and create permanent changes passed through generations. At present, research into human germline editing for space remains speculative, debated heavily but not pursued in practice. Technical obstacles support that caution. CRISPR is precise compared to past tools, but unintended, off-target changes still occur. On Earth, sequencing platforms can identify those errors quickly. In orbit, oversight is constrained by limited resources. Missed mistakes could accumulate until failures become evident in failing crops or sick astronauts. Even delivery methods are complicated when gravity no longer distributes fluids normally through tissues. Before any shift from microbes to human cells, these problems must be addressed. Current yeast and bacterial studies are the first rung of a long ladder, not a template for imminent colonist modification. Biosecurity risks widen the conversation. The ability to design resilient microbes or edit genes to resist damage also means the potential to create pathogens unsuitable for a closed environment. A harmful edit, intentional or accidental, could spread through habitats with no easy containment. Portable genetic tools are already cheap and accessible on Earth. In a space station or settlement, containment options diminish drastically. Safety protocols to prevent malicious or careless misuse are essential before expanded adoption. Even with caution, the data now returning from space highlight possibilities unimaginable a decade ago. Organisms edited for enhanced DNA repair show responses under orbit conditions never captured on Earth. Some recover more effectively from radiation strikes, others reorganize cellular systems in unpredictable ways. These signs suggest that life encodes greater adaptability than previously assumed. Adaptability gene editing may be able to coax into reliable resilience. Each deviation from expectation is more than an anomaly. It is a clue to how biology might extend its reach beyond the planet where it began. The broader picture is that sustaining colonies will depend on whether such adaptability can be expanded to crops, supporting microbes, and eventually human physiology safely. Without it, habitats remain temporary constructs propped up by costly supply chains and fragile infrastructures. With it, the prospect of people enduring in hostile environments draws closer to reality. But the cost is a reevaluation of how freely humanity alters the very code that defines its species. Would you sign up to be modified from Mars? That's not a sci-fi thought experiment anymore. It's an ethical debate scientists are having now. As the debate unfolds, one theme becomes clear. Space has made gene editing a question not only of repairing damage, but of redefining what roles organisms, including ourselves, are expected to play. And back on Earth, those expectations are already shifting in subtler ways, where the organisms being edited are not crops or microbes, but the animals millions of people invite into their homes. The weird world of designer pets. Pets carry a different kind of weight in this conversation. They are not anonymous organisms in fields or microbes in space. They share our homes, shape our routines, and often take the role of family members. Yet even here, gene editing is beginning to reshape possibilities. The odd-sounding but very real category of designer pets has moved from scientific curiosity to early-stage prototypes. Research programs in Asia and the United States have already produced micropigs adapted to stay small, as well as cats edited to reduce the proteins that typically trigger allergic reactions in owners. These examples are still limited to controlled labs and small pilot projects, but they hint at a market where animals are tailored to human preference rather than bred along older lines. The first public glimpse of this trend came several years ago when Chinese labs produced mini-pigs through edits similar to CRISPR. Initially designed as models for human disease, the pigs drew attention because their small size made them candidates for household life. At the same time, companies explored editing cats to suppress FeLD1, the protein in their saliva and dander that sparks allergic reactions in humans. Unlike these proof of concepts, consumer availability remains narrow. Many edited animals exist only as research demonstrations, and regulations vary widely between countries. Still, their very existence signals pressure on markets to move in that direction. Companies see opportunities in what some now call status pets. The logic is familiar. If consumers already spend on rare breeds, exotic appearances, and premium products, then an animal edited for uniqueness or convenience fits that same market logic. A hypoallergenic kitten could make cat ownership accessible to millions who would otherwise face health barriers. A permanently small pig promises novelty in settings where regular pigs would not be sustainable. Even glow-tinged fish and reptiles, intended as classroom tools or curiosities, are marketed as novelties. But availability remains constrained, not mass-market catalogs, but prototypes, luxury offerings, or pilot sales in permissive jurisdictions. On one side, creating pets that reduce barriers to ownership seems like a genuine benefit. If allergies no longer drive families to abandon animals, both humans and pets gain stability. On the other, critics argue that novelty traits risk crossing into vanity. When the primary appeal is uniqueness or prestige, tiny pigs, glowing fish, or modification for color, it becomes hard to separate genuine welfare improvement from indulgence. Even convenience edits can carry trade-offs. Animal welfare experts caution that removing allergens may also alter important immune functions, or that constraining growth in pigs could leave them with metabolic stress later in life. Edits that seem harmless in theory can still impose costs that only surface with time. Companies press ahead nonetheless, branding these projects as luxury options. Experimental hypoallergenic kittens are introduced as premium products with waiting lists. Micropigs appear in marketing as gentle companions, often alongside accessories and services, framing them as high-status lifestyle choices. Investors see profit potential because pet industries already generate hundreds of billions globally. In urban centers where pets are closely linked to identity, the draw of a rare or engineered animal can be strong. This creates a fast-moving luxury market that often outruns ethical debate and leaves regulators struggling to keep pace. The global legal framework is inconsistent. China has issued rules limiting how edited animals may be sold, recognizing both scientific potential and ethical risks. In the United States, regulation is patchier, often borrowing language from agricultural policy or held biotech not suited for companion animals. In Europe, skepticism rooted in GMO controversies carries over into household animal debates. This patchwork means an edited pet could be legal in one territory while banned in another, creating loopholes where companies establish production in permissive regions and export elsewhere. Regulation lags while demand grows, and consumers may not even know whether a pet they adopt already carries edits. Public opinion reflects the same unevenness. In some countries, any hint of genetic editing raises resistance, often linked to previous controversies over modified crops. Elsewhere, curiosity dominates. Videos of unusual pets spread online, attracting fascination that obscures questions about risk. For some, the very strangeness is a selling point, reinforcing the status angle. For others, the edits spark anxieties that human desires are shaping animals in directions with little regard for their welfare. Governments, meanwhile, face pressure to respond, sometimes lumping pets into broad restrictions to avoid controversy. The ethical divide often comes down to whether this is seen as an extension of selective breeding or a departure from it. For centuries, humans have crossed and selected for traits: flat-faced dogs, long-haired cats, short-legged corgis. Many of those traits carry health consequences far worse than what gene editing currently proposes. Advocates argue that gene editing might allow healthier outcomes in place of painful selectively bred traits. Critics respond that editing accelerates human intervention without the buffer of natural variation, and that the temptation to satisfy demand will outweigh cautious welfare checks. Either way, scrutiny of animal outcomes becomes critical before widespread acceptance. If you could choose any genetic trait for a pet, what would it be, and should you? The question may sound speculative, but it reflects real pressures now playing out in labs and markets. Some might ask for a cat that never sheds, others for a dog immune to common diseases. Each possibility illustrates both the attraction and the unease. Making animals conform more perfectly to human lives while removing aspects that once made them challenging, unpredictable companions. From a business perspective, experiments with pets look less like fringe science and more like an extension of existing consumer markets. Around the world, households already invest in premium feeds, clothing, and wellness products for animals. A genetically customized pet becomes one more layer of personalization, but animal welfare groups warn that this frame risks turning living beings into consumables. A product-like view overlooks the reality that every pet has needs beyond the trait it was edited for. Welfare concerns rarely sit comfortably next to luxury marketing strategies. The uneven geography of technology highlights another layer. In wealthier cities, designer pets might circulate among households, emphasizing lifestyle or novelty. In lower-income regions, where animals serve working roles or companionship without luxury framing, these developments can feel remote, yet global narratives still center on them. That imbalance mirrors broader technology adoption patterns, where luxury products for the privileged dominate the conversation even when basic needs elsewhere remain unmet. As of 2025, edited pets remain mostly research projects, prototypes, and pilots, not yet mainstream. Regulation is not caught up, and oversight frameworks built for agriculture do not yet map onto living room animals. But the line is moving. If markets push ahead without coordinated standards, households could face waves of new animals carrying edits with little data on their welfare, health outcomes, or long-term care needs. That scenario presses governments, companies, and consumers to weigh not just what is technically possible, but what is socially acceptable. At the heart of the debate lies the same tension repeating throughout gene editing. When convenience or novelty becomes possible, how do we decide whether it should be pursued? Designer pets drive that tension close to home, literally within household walls, bound up with emotions and affections. But they are also a rehearsal for larger questions. If society normalizes editing animals for lifestyle, the next frontier is far more consequential. What happens when the subject of editing is not a cat, a dog, or a pig, but ourselves? The ethics of making humans better. If gene editing has already allowed scientists to repair defective blood cells inside patients and correct dangerous mutations in the body, the shift toward using the same methods for something beyond disease begins to feel less like science fiction and more like an obvious next step. The question is no longer whether the tools could be applied to traits like muscle strength, height, or cognition, but whether society should permit that application at all. In 2025, conversations about enhancement hang over the field like a shadow, impossible to ignore, yet often unspoken in formal settings. Enhancement appeals precisely because it promises more than healing. Instead of preventing inherited illness, it suggests improving traits that are already healthy. Sharper memory, quicker reflexes, or immune systems that shrug off routine infections. In a culture already accustomed to elective treatments for appearance, fertility clinics, and performance drugs, the leap to genetic fine-tuning feels like a continuation rather than a rupture. Parents who spend large sums on IVF or pre-implantation genetic screening may start to see editing as another option to stack the odds in their child's favor. The line between preventing disease and designing advantage is not written in the DNA itself. It is drawn by the choices of people, regulators, and markets. The figure who brought this debate into public consciousness remains Ejanqui. In 2018, he revealed that twin girls had been born following edits intended to make them resistant to HIV. His announcement triggered worldwide condemnation, not for lack of imagination, but because he bypassed safeguards, ignored consent norms, and imposed irreversible changes on future generations. The backlash was swift. He was investigated and imprisoned in China, barred from research, and his work was denounced as reckless. Importantly, the scandal did not end discussion. It reinforced the urgency of global coordination. His case crystallized why germline editing, the kind that alters embryos, eggs, or sperm, and passes those edits to descendants, provokes the deepest concern. Ejanqui's claim led to renewed calls for international moratoria and strict regulation. It also showed one determined actor could bypass consensus and cause lasting consequences. To understand why germline experiments cut so deeply into ethical debate, it helps to distinguish them clearly from somatic intervention. Somatic edits affect only the person treated. They stop at the body's edge. A therapy editing cells inside an adult with blood disease or hereditary blindness is considered somatic. By contrast, germline edits change the foundational DNA of embryos or reproductive cells, embedding modifications into future lineages. Their permanence affecting children not yet born is what drives fierce warnings from organizations like the WHO and the UN. These bodies have stressed that while somatic therapies are steadily advancing into medical clinics, germline editing demands far stricter caution because one mistake echoes across generations. Despite public condemnation, interest in pushing further has not vanished. In Russia, geneticist Denis Rebrikov openly declared intentions to pursue germline modifications, including edits to prevent HIV transmission. While his proposals stalled under scrutiny, they highlight how researchers can operate in gray areas when countries adopt divergent rules. The fragmented regulatory environment means some governments prohibit embryo editing outright, while others permit limited research under opaque conditions. Without a single global authority, enforcement is uneven. A regulated trial in one region may amount to a banned experiment in another, leaving opportunities for experiments to migrate across borders or into jurisdictions more willing to tolerate them. This unevenness shapes public trust. Surveys consistently show strong support for therapeutic editing that prevents devastating illness. Genetic interventions for Huntington's disease, cystic fibrosis, or severe metabolic disorders are seen as humane, even necessary. But when questions extend to traits not tied to suffering, intelligence, height, physical beauty, support falls sharply. The shift is less about biological feasibility than social fairness. Saving a child from a fatal disease is widely regarded as just. Granting a child superior abilities only accessible to the wealthy is far more contentious. The distinction illustrates why inequality looms so large over this conversation. Current CRISPR therapies in clinical stages already carry high costs. Experimental treatments often exceed $1 million per patient. If enhancements emerge through similar technology, they would be available first to the wealthy. Two families entering the same fertility clinic might walk out with different options, one with only basic screenings, the other with access. Edits promising stronger children. The result would extend disparity from education and healthcare into the fundamental biology of future generations. Critics warn of society stratified not just by money but by design genetic differences. Some argue that enhancements could one day be distributed broadly, subsidized by states seeking healthier or more capable populations. In theory, governments might use public programs to democratize access and prevent inequality. From organ transplants to advanced cancer care, new interventions typically remain out of reach for poorer regions for decades. If nations with resources introduce enhancements while others cannot, inequality becomes international. It would not be one society split by access, but a planet divided by biology. Outside official labs, the democratization of tools raises another challenge. CRISPR reagents are relatively cheap, and basic protocols circulate freely online. A growing community of do-it-yourself biologists experiments with microbes, plants, and sometimes animal cells. Most projects remain minor, but the leap toward human editing is less unimaginable with each year. Regulators worry not because basement laboratories are already producing human-engineered embryos, but because the barrier keeps dropping. A tool powerful enough to extend life or cure disease in experienced hands could also be misapplied by hobbyists or fringe groups seeking enhancement for spectacle or ideology. The tension between therapy and enhancement echoes into every corner of policymaking. Should international law prohibit germline enhancement outright while allowing tightly controlled somatic therapies? Should there be categories of edits distinguished by function, disease prevention allowed, cosmetic alteration banned? Different nations answer differently. The United Kingdom permits limited embryo research under licenses. The United States restricts funding and maintains ban-like barriers. China, despite their He Jiankui scandal, allows some embryo work under defined oversight. Attempts at global declarations have urged transparency and responsibility, but without binding authority, enforcement remains inconsistent. Critics emphasize practical safety alongside abstract ethics. CRISPR is more precise than earlier tools, but no method is flawless. Off-target effects, unintended mutations at places not targeted, still occur. In embryos, such mistakes replicate into every cell as the organism grows. The danger is not only an unplanned outcome, but also harm emerging decades later in ways no trial could anticipate. Advocates of enhancement seldom present an equivalent medical justification for offsetting those risks. Extending life expectancy by avoiding cystic fibrosis differs morally from selecting taller stature. Regulators note this difference when drawing boundaries. Military and commercial pressure complicates matters. Enhancements promising resilience to radiation or resistance to fatigue are tempting to defense organizations. Corporations foresee lucrative markets among parents vying for competitive advantage for their children. Once positioned as an arms race, enhancements acquire urgency. If one state pursues them, another may feel compelled to follow, whether for athletes, workers, or soldiers. In that environment, secrecy is not just about avoiding scandal, but about racing for advantage. He Jianqui's actions underscored this. Once one person crosses the threshold, the theoretical frontier becomes material. Should the freedom of parents to reduce their child's risk of disease outweigh risk of altering the human germline permanently? Or should society prohibit germline editing altogether until global safety and fairness can be ensured? Somatic therapies continue moving steadily toward widespread medical use, giving real patients relief today. Germline efforts, by contrast, promise uncertain advantages, but carry risks to descendants who cannot consent. That contrast forces a straightforward choice: treat defects one body at a time, or attempt to remake heredity itself. Behind these questions lies a broader cultural unease. Editing humans for advantage raises the possibility of redefining what counts as normal. If unaltered children compete against those chosen for optimized traits, will society see some lives as lesser? Will equality shift from being a political and economic issue to a biological one? The scenario unsettles because it reframes what it means to belong. The same editing tools capable of sparing children from cruel disease could also transform human diversity into a marketplace. Whether humanity treats those possibilities as acceptable opportunity or unacceptable hubris remains undecided. He Jianqui's case exposed the fragility of consensus. Global outrage in a prison sentence stopped one rogue scientist, but enforcement built on. Outrage alone cannot hold indefinitely. The tools are cheap, the expertise is spreading, and motivations, financial, military, or parental, are powerful. The present may still separate therapy from enhancement, but that separation is not guaranteed to endure. And that brings the question directly to you. Should we prioritize a parent's right to eliminate suffering through therapy, or draw firm lines against enhancements that alter the human line for generations? Where do you fall in that spectrum? Tell us which side you lean toward. The challenges raised by human editing point to something larger still. Debates about disease, fairness, and inequality highlight the limits of how we manage change. But scientists are also applying gene editing beyond present and future individuals, turning the same tools toward organisms of the distant past. The implications reach even further into how we write and rewrite genetic material as though it were code waiting to be recompiled. From woolly mammoths to living code. When scientists discuss the return of the woolly mammoth today, it is not framed as a simple act of resurrection. Instead, it is presented as an experiment in ecological engineering, an attempt to explore whether mammoth-like traits engineered into elephants might help reshape cold ecosystems and even slow the thawing of permafrost. Figures such as George Church have become well known for advancing the idea, building elephant hybrids that carry traits like thick fur, fat layers, and cold-tolerant hemoglobin. The stated goal is not to recreate a genetically identical woolly mammoth. It is to edit a modern relative in ways that echo traits of the extinct animal, then test whether those traits could be applied to landscape management and climate mitigation. Technically, this takes the form of piecing together ancient DNA fragments, aligning them with living elephant sequences, and identifying genes that once encoded code adaptations. Where genetic instructions are intact, CRISPR or related editing tools can be used to insert them into elephant cells. The project is not science fiction. Embryonic stem cells carrying these edits already exist in controlled labs, and research is ongoing to shift those edits into viable embryos. The offspring will not be mammoths in the historical sense. They will be elephants engineered to carry select mammoth traits. The prospect is less about bringing back an animal lost to the Ice Age, and more about testing the limits of whether genomes can be reconstructed across time, with fragments of ancient code integrated into present-day hosts. Viewing DNA as editable code reconfigures how scientists, companies, and the public think about biology. Instead of permanent loss, extinction begins to resemble a kind of archive, where traits can in principle be recompiled. A mammoth's ability to grow long fur becomes just another instruction in that archive, able to be paired with elephantine immunity or fertility traits, each edit spliced and measured. The possibility extends far beyond mammoths. Any preserved sample, from museum drawers, amber, frozen soils, or seed banks, represents potential source material. An extinct species becomes not only an absence in history, but also a toolkit of recoverable instructions. That shift from inheritance to design language opens doors that previous generations assumed permanently locked. The framing of organisms as modular systems parallels computer software, where lines of code can be borrowed or rewritten across projects. Mammoth projects demonstrate this in practice. No one expects the outcome to be a perfect replica of an extinct animal. Instead, the goal is a working hybrid, an elephant that grows insulating fat, produces fur, and tolerates Arctic winter. Calling it a mammoth may capture imagination, but technically it is an edited elephant carrying selected traits. The distinction matters because the aim is deliberate and functional. Evaluate whether such a hybrid could survive in tundra conditions, reshape grasslands, and in theory manipulate the carbon balance of permafrost regions. All of this has been made possible by collapsing costs. Sequencing once required billions of dollars, as in the Human Genome Project, and editing DNA was slow and imprecise. Today, a full human genome can be sequenced for hundreds of dollars, and editing segments has become routine. This shift has lowered entry barriers, multiplying the number of labs and startups involved. What once was the province of elite institutions now extends to new companies across continents, each applying the same tools to projects in conservation, medicine, agriculture, or consumer goods. The price drop, especially for sequencing and CRISPR editing, has been likened to the leap from hand-built cars to Ford's Model T. Accessibility grows, and activity rapidly multiplies. Alongside conservation projects comes a parallel transformation of biotechnology into something resembling a service market. Synthetic biology firms now advertise DNA blueprints as if they were downloadable applications. In this framing, a laboratory no longer has to engineer a gene edit from the ground up. Instead, it browses a catalog of standardized sequences, from disease-resistant traits for crops to targeted metabolic functions in microbes. These files can then be combined or adjusted, much like pre-written code pulled into a program. The economic logic is familiar. Standardization reduces costs, accelerates timelines, and shifts the field from specialized artisanal work into scalable production. Comparisons to an app store are tempting, and they clarify why access is spreading so quickly, but they come with caveats. Digital programs execute within controlled systems. Biological code interacts with complex environments where consequences are unpredictable. An edit that produces healthy alterations in one host may cause stress or harmful mutations in another. Unlike buggy software that can simply be updated or deleted, genetic alterations spread through living systems and can persist indefinitely. It is this gap between metaphor and reality that drives both enthusiasm and alarm. DNA can be treated like code, but biology does not respond with the same predictability. The Mammoth Project crystallizes these tensions. Supporters argue that hybrid elephants re-engineered for cold climates could serve as tools of ecological restoration. Grazing herds might stimulate the growth of grasses across tundra, slowing permafrost melt and potentially reducing carbon release. It is a vision that positions gene editing as a lever for climate mitigation. Critics see misplaced priorities. They argue that resources diverted to experimental hybrids could instead protect living elephants, combat poaching, or restore existing habitats. Critics also raise concerns that enthusiasm for de-extinction may reduce urgency around conservation, spreading the dangerous idea that loss is temporary because future science can reverse it. These critiques draw on concrete ethical concerns. A hybrid released into tundra would interact with predators, competitors, and plants in ways that models cannot fully predict. Ecologists warn that edited reintroductions may behave like invasive species, disrupting food webs and outcompeting native animals. The very traits that make hybrids resilient could destabilize fragile ecosystems. At the same time, diverting attention and funding from endangered species shifts conservation away from preventing extinctions today. Experts underline a central point. Critics worry resources for de-extinction could be better spent on protecting currently endangered species and habitats. Intellectual property adds complexity. Many companies claim ownership over synthetic sequences, even when those sequences derive from natural organisms. A DNA blueprint that encodes disease resistance may be packaged and licensed, despite being a recoded version of traits found in wild plants or animals. This sparks familiar debates about whether genetic resources are common heritage or proprietary goods. The issue mirrors earlier controversies over seed patents and genetic medicines, but is now expanded by the speed and scale of CRISPR. Who owns redesigned traits, labs, corporations, or humanity, is now a pressing policy question. The lower costs also democratize risks. Inexpensive kits make gene editing accessible to schools and individual hobbyists. Paired with companies selling pre-packaged sequences, this makes powerful tools widely available. On the one hand, innovation accelerates. Independent labs can create microbes to degrade plastics or plants that signal stress with visual cues. On the other hand, the same accessibility invites accidents or misuse. Off-target edits could destabilize organisms, or poorly understood sequences could proliferate in ways difficult to control. Regulators face the challenge of preventing harm while not stifling innovation, a balance yet unresolved. Culturally, the framing of DNA as code taps into broad expectations about technology. Investors embrace it, startups promote it, and public imagination is drawn to scenarios where apps aren't downloaded onto devices but written in the living organisms. Yet genetic code is not software. It is entangled with unpredictability, shaped by environment, evolution, and context. Every blueprint imported into an organism carries potential for emergent outcomes. Simplifying biology into code risks encouraging expectations of reliability where none exists. This does not erase the potential, but it requires a steady reminder. Biology resists control more stubbornly than engineered machines. Nonetheless, momentum gathers. Startups raise capital to design microbes that manufacture textiles, plants that perform environmental cleansing, and bacteria that produce consumer chemicals. Yeast has already been adapted to make vaccines and biofuels, signposting what may follow. For advocates each, success constructs another proof of principle. Treat DNA as a set of instructions, and organisms can be customized for new roles. For critics, every step blurs the line between respecting life and reducing it to a disposable product. Both positions acknowledge the same reality. Biology is increasingly treated as a programmable substrate. A future where DNA catalogs resemble an app marketplace is easy to imagine. A farmer orders resilience against drought with a download. A biohacker chooses color traits for ornamental fish. A Martian settlement requests blueprints tailored to soil chemistry and radiation. Each scenario extends biology in the service design. The danger is that organisms become valued primarily for utility, leading society to treat them as interchangeable platforms rather than living beings. That shift, ethical as much as practical, reframes the human relationship with other species. The Mammoth Project thus becomes emblematic not for its spectacle, but for the broader lesson: extinction is no longer final. Ancient genes can be retrieved, reshaped, and reintroduced into living systems. This possibility represents both scientific ingenuity and cultural risk. The temptation to solve biodiversity crises with re-engineering could distract from the harder work of preventing loss in the first place. Is the extinction a worthy scientific tool or a dangerous distraction from stopping extinctions now? The question is unresolved and the debate continues. What is certain is that the way we approach DNA, as archive, as commodity, as toolkit, is rewriting biology itself. The woolly mammoth may or may not return in any recognizable form, but the act of decoding its traits and inserting them into modern life proves that genes travel across time as easily as across species. That transformation carries implications for conservation, for commerce, and for identity. The implications of programmable life extend everywhere. From climate projects to consumer markets, from ancient tundra to modern clinics, gene editing is no longer confined to the realm of speculation. It is already pressing on the systems that feed, shelter, and define us, systems whose stability touches everyone. The unresolved question is how far societies will allow the logic of code to reshape living systems. Gene editing is no longer confined to laboratory experiments. It is shaping the food on supermarket shelves, the way we manage livestock, and the pets that enter households. In medicine, progress has been even faster. 2025 saw the first personalized CRISPR therapy delivered to an infant with a rare metabolic disorder at Chop and Penn, while base and prime editors are advancing precision in early trials. Real progress that underscores how quickly this field is moving. If there is one takeaway, it's that gene editing is no longer hypothetical. Its influence will touch healthcare, agriculture, and policy within a decade. These tools can cure and enhance, but the difference will be defined by ethics, regulation, and collective choice, not technology alone. So what about you? Would you trust gene editing most in medicine, conservation, agriculture, or not at all? Share your thoughts in the comments, and if you want more balanced deep dives on this future, consider subscribing.